Well, thank you. It's a pleasure to be here with you this morning and to talk to you about the wonder of DNA. Now, there is so much information that we could talk about when we're dealing with the complexity that we find in DNA that it's kind of difficult to figure out what to put into a 1 hour presentation. So we're going to kind of be looking at this from about 30,000 feet and without going into extensive detail on any one topic, but hopefully gaining an appreciation for a very small fraction of the complex design that we find in DNA. And we're mainly going to talk about human DNA because that's where most of the research has been done. And obviously, that's DNA that's very important to us. So that's what we're going to focus on today. Now, I want to begin by talking about how the human DNA is organized. So I'm going to kind of give you a little bit of background here because some of these terms you'll hear come up over and over in the presentation. So just some basic facts about the human genome. The genome is the DNA inside the nucleus of our cells. In humans, it is composed of about 3 billion base pairs that form a twisted double helix. And so there's two strands of bases that pair with each other, and we represent those bases with 4 letters, A, C, T, and G. And so most of you have probably seen an image very similar to this one of the twisted double helix, and there are rules, so to speak, that have to be followed in DNA. A always binds to T, and C always binds to G to form that double helix. Now, in the human genome, it's at least organized the DNA into 23 pairs of chromosomes. Now, in those chromosomes is only about 20,000 genes, and genes are the instructions for the proteins, okay? That's what makes up our body. That's what gives it structure, gives it function. And that's only about 2% of our DNA. It's a very small fraction. The other 98% has been affectionately termed junk. Right? And we're going to talk about that because it turns out that's not a very good term for it. Now, I'm going to show you a short video that kind of will hopefully give you an idea of the size of the genome and its organization.
Think of how many letters fit on a single page. And now imagine a stack of pages 300 feet tall. That's how much information is stored in the DNA inside every human cell, the entire human genome. If you sort through the 3 billion letters that make up the human genome, you find some surprising things. Only about 1% of the 3 billion letters directly codes for proteins. Of the rest, about 25% make up genes and their regulatory elements. The function of the remaining letters is still unclear. Some of it may be redundant information left over from our evolutionary past.
Okay, now there's a couple of things I disagree with in this. One is that I would disagree that that stack that you see there is how much information is stored in the DNA. Rather, that stack represents if you wrote out our DNA with letters, okay, A, C, T, and G, that's what you would get, okay? You would get that 300-foot stack of papers. But that doesn't represent how much information is stored there because DNA is a 3-dimensional structure, as we're going to talk about. And as we're going to see, there's layer upon layer upon layer of information there. Now, the narrator also says that towards the end that a lot of the DNA, which they kind of call non-coding DNA, is redundant information left over from our evolutionary past. So in essence, she's saying that most of our DNA is junk, right? Even though it's unclear what the function is, she says, they don't think it's important. And we're going to talk about that a little bit later. Now, how is the information that's in the DNA utilized to maintain and build living things? Because obviously, you have to get from the instructions to the final product. So how do you do that? And for that, we look at what we call the central dogma of molecular biology. And so we have DNA, and it is transcribed into RNA, which kind of is like an intermediate carrying that information. And then that is translated into a protein. So transcription and translation are the 2 major ways that we get from the instructions to the final product. So that's the way in which the DNA is expressed, okay? It doesn't do any good just to have the instructions. You have to have a way to build off those instructions, so to speak. How do you build the final product? And while it may look very simplistic on the screen, it is a very complex and very highly regulated process. And so this next video, again, is going to take a rather simplified look at how we get from the DNA to the protein.
Here is a cell, the basic unit of all living tissue. In most human cells, there is a structure called the nucleus. The nucleus contains the genome. In humans, the genome is split between 23 pairs of chromosomes. Each chromosome contains a long strand of DNA, tightly packaged around proteins called histones. Within the DNA are sections called genes. These genes contain the instructions for making proteins. When a gene is switched on, an enzyme called RNA polymerase attaches to the start of the gene. It moves along the DNA, making a strand of messenger RNA out of free bases in the nucleus. The DNA code determines the order in which the free bases are added to the messenger RNA. This process is called transcription. Before the messenger RNA can be used as a template for the production of proteins, it needs to be processed. This involves removing and adding sections of RNA. The messenger RNA then moves out of the nucleus into the cytoplasm. Protein factories in the cytoplasm, called ribosomes, bind to the messenger RNA. The ribosome reads the code in the messenger RNA to produce a chain made up of amino acids. There are 20 different types of amino acid. Transfer RNA molecules carry the amino acids to the ribosome. The messenger RNA is read 3 bases at a time. As each triplet is read, a transfer RNA delivers the corresponding amino acid. This is added to a growing chain of amino acids. Once the last amino acid has been added, the chain folds into a complex 3D shape to form the protein.
Okay, now that again is a very simplified version of the processes. Okay, there's a lot more going on, trust me, a lot more proteins, a lot more things floating around that you don't see. But remember, this is only true for about 2% of our DNA, okay? That goes, because again, that's only 2% has genes. So what is the other 98% doing? And what we're finding out is that a lot of that 98% is involved in regulation, okay? Because these processes have to be highly regulated. Because remember, all the DNA is in every cell of your body, right? But you need brain cells to produce brain proteins and liver cells to produce liver proteins, right? You can't have all the DNA expressed in every cell. They need to be specialized. So there's a lot of regulation there. So you need to know when to produce the proteins, how much, why, under what condition. It's going to be different if you're talking about a baby that's growing in the womb versus a child versus an adult versus an older adult. There's going to be all this regulation that's needed even throughout a person's lifetime to regulate these proteins properly. So what we're going to be talking a lot about today is where that regulation resides, because I think that regulation is a really great example of the design and the complexity that we see in DNA. And so I hope that you'll see, again, just a very tiny fraction of the beautiful and amazing design that's there. And as we look at the complexity and the design of DNA, which really falls into the category of observational science. That's what we're going to be talking about a lot today. These are things that we observe in the present. They're in the here and now. They're observable, they're testable, they're repeatable. And so we need to keep in mind, though, that who or what we attribute that complexity that we observe in DNA to depends on our starting points, okay? What is our worldview for looking at DNA? That falls into the category of historical science, how the DNA got here, so to speak, how it got so complex. And so because the origin of that complexity that we observe is an event that has happened in the past. We can't observe it, we can't test it, and we can't repeat it. So it very much depends on our starting point for how we think that complexity came to be. Now, evolutionists believe that the complexity that we observe in DNA is not designed, but rather apparently designed. Okay? That's the term that they'll use. So it looks designed, but it really isn't. And again, that's based on their starting points for knowing about the past. They believe man's ideas about the past, that all life, including DNA, that's part of that life, has come about by random chance over millions of years. Leslie Orgel, a British chemist, said this: "It is genuinely surprising that an organism that has evolved by random mutation and selection appears to be designed. The idea is contrary to intuition. It is true, but to many, it will always remain ridiculous." And why is it ridiculous, right? Why is it contrary to intuition? Because when you look at something that's that complex, you expect it to have been designed by something, right? I mean, think about it. We look at a computer, we look at a phone, we would never say those things came about by random chance. They're complex, they have to have a designer. Yet these people are looking at the human body, which is infinitely more complex, and saying it came about by random chance. Okay, it is very, very contrary to intuition. So why do this? I mean, why do evolutionists rule that possibility out from the start? Why is it not really designed? And I think these two British astronomers, Hoyle and Rick Remanescing, sum it up very nicely. They say to involve purpose is, in the eyes of biologists, the ultimate scientific sin. The revulsion which biologists feel to the thought that purpose might have a place in the structure of biology is therefore a revulsion to the concept that biology might have a connection to an intelligence higher than our own. Because if something is designed, right, if it does seem to be there for a purpose, that means it's not come about by random chance. That means it's come about by a designer. And so as we study DNA today, what we're going to see is that its design confirms and is consistent with historical science that based on God's word, and God is the creator and designer of our DNA and his intelligence, and is inconsistent and does not confirm that DNA is a result of random chance over evolutionary eons of time. Now, being a geneticist, from my personal perspective on this, and understanding and learning and teaching about the amazing design that's DNA, it makes me realize even more that this is not an issue of the evidence, because the evidence is crystal clear, okay? It truly is an authority issue, and it ultimately comes down to a heart issue, that many scientists want to be their own God instead of believing in the one true God who designed and created our DNA. So today we're going to talk about two major types of design that I have sort of categorized them into. And the first one I'm going to call sequence design, which has to do with the actual sequence of DNA. And then we're going to talk about something called structural design, which has to do more with the three-dimensional shape of DNA. And again, even within these, I'm only discussing a very small part of the design that we see there. So, all right, sequence design, the actual sequence of the DNA. So the first thing we're going to talk about here is something I've termed hidden codes. Now, this does not mean messages from God in your DNA, okay? They're not there, right? It does not mean Bible verses in your DNA. Why would he do that, right? He's given us the Bible. We have that. We don't need to decode DNA to find that out. But there are certain codes, I call hidden codes there, because we didn't know they were there. We didn't know these things were coding for things, and so that's why I call them hidden. So the first one we're going to talk about is something called douons. Now, douons are codons found in genes. Now, let me explain what a codon is. In your DNA, in the part that codes for protein, the genes essentially are composed of three-letter words. Those three-letter words are called codons because each codon codes for a particular amino acid in the final protein. So, for example, GCA, which is the first codon in the DNA here, codes for the amino acid alanine. And then the next codon, AGA, codes for the amino acid arginine. Okay, so that's how you go. It's just three-letter words, and they all mean something basically for the final protein. So there are 64 possible combinations of the four bases of DNA, A, C, T, and G, into these three-letter words. And there are only 20 amino acids. So there's what we call overlap, basically. So some codons, like you see here in this table, scientists have figured out which codons code for which amino acid. These 4 codons that you see here all code for the amino acid serine. Doesn't matter which of those you have, you get the same amino acid. These 4 all code for the amino acid threonine. So most scientists used to think that even if you got mutations that gave you like a different codon, as long as you got the same amino acid, everything was okay. So for example, for serine, you could get TCT, or if there was a mutation that occurred and you have TCA, it's okay, right? Because you still get the same amino acid out of it. Well, that's the assuming that codons only do one thing, right? Which is code for the amino acid. And as it turns out, they don't. They do multiple things. And so they have what we could say dual purposes, which is why they're called, these particular ones are called duons. Not only do they specify the amino acid in the protein, but they also specify where factors bind to and regulate the expression of the gene. So they're involved in regulation, which again, this is totally, no one ever thought this before. We just thought they coded for the amino acid. Now they're involved in regulation, okay, from going from DNA to RNA, and that this applies to about 15% of human codons. So they're doing 2 things. Now, that's going to be a problem for evolution, and it's really twofold. Because first of all, how do you explain the origins of these duons by random chance mutations? So not only do you have to have in the DNA a codon to code for the correct amino acid for the protein, but also has to regulate the expression of that gene correctly, like how fast it's going to go, or when it's going to happen, or under what conditions it's going to happen. So because it's doing 2 things. You have to evolve 2 things simultaneously. So I always say evolution got extremely lucky, right, in order to do this, right? I mean, if it's just doing 1 thing, you might say, well, okay, maybe that could happen by chance, but 2? All right, now we've just compounded the problem. And I'm going to guess that they probably do more than 2 things. This is just all we've discovered so far, okay? And the other thing is what are the odds that a mutation is going to be beneficial for both functions? Because that's how evolution works, right? Is by random chance mutation. So what are the odds that even if you end up with the same amino acid, right? So it's okay, you still got the same amino acid, but what about for the other function, right? The regulatory function. Oh, well, now you've messed that up. So it does matter which codon's there, right? You can't just take your pick. You have to have certain ones there because they're not just doing 1 thing. So it would be very hard to believe that that mutations would be beneficial for both functions. Even if it would be okay for one, it would probably likely destroy the other function. So you don't have the gain basically that you need for evolution. So it really becomes even more implausible to believe that random mutations over time have generated the complexity and the diversity of organisms that we see today. Because again, you're having to evolve 2 things at one time. This is clearly design, is not disorder. And one of the researchers that was involved in this research said, for over 40 years, we have assumed that DNA changes affecting the genetic code solely impact how proteins are made. That's the only thing it affects. Now we know that this basic assumption about reading the human genome missed half of the picture. You know what I'm going to guess is they've missed 99.9% of the picture, okay? Because we still haven't figured all of these things out yet. And why did they assume it was so simplistic, right, to start with? Well, because again, they're starting with evolutionary assumptions that would predict relative simplicity because everything came about by chance from a simple common ancestor. The level of complexity that we're seeing here is inconsistent with what they would predict, but totally consistent with what we would predict starting with God's word and God as a designer and creator of our DNA. Now let's look at another hidden code because there's other ones. And I've termed these translational pausing codons, TPCs. Now they're also codons found in genes, but they're not involved in regulation. They're involved in something else. They have a dual purpose. They specify the amino acid. Okay, that hasn't changed. But now these codons allow for what we call pausing during translation. So they're not involved in transcription like we just saw. Now they're involved in translation for the proper folding of the protein. Now let me illustrate what I mean by this. When I showed this video earlier, like I said, this is really simplistic. And they're saying, oh, the protein is formed and then it folds up into the correct structure. Technically, that's not how it happens, okay? So as it's being formed, it's actually folding. That is important. If it waits until the end, it most likely will fold into an improper structure. And if you don't have the right structure, you don't get the right function. So it's really important that it fold as it goes. So sometimes as it's basically feeding through the ribosome here, there's some codons that take longer to find a match for, essentially. And so it pauses. It kind of stops it for a little bit, and that helps it fold correctly, that protein. And then the match will be found, and it will continue on. And then another one might come up, and it takes a while to find a match, so it pauses, and then it folds a little bit more, because these proteins can be really, really big. Again, that's like a really short one they're showing you there. They can be 300s or thousands of amino acids long. So you've got to be able to do this. Now, so again, they think they found this to be really important for the correct folding for the protein, and that is very important for function. If you don't have the right folds, you don't have the right function. Now, again, this is going to be a major problem for evolution, very much like we saw for the duons, because how do you explain the origin of these TPCs by random chance mutation? Because now not only do you have to code for the correct amino acid, but you have to have a codon that's such that it pauses during translation so that you get the right fold. Right? I mean, how does it even know this to evolve this, right? Oh, well, I need to pause here so this fold is correct, so this protein can do this. Evolution does not have a mind, okay? And so that's definitely going to be a problem for it. So evolution would have had to got extremely lucky to be able to do this. And again, what are the odds that a mutation is going to be beneficial for both functions? Even if you're still okay on the amino acid side, you're probably not okay on the pausing side. And so it's not going to have the right structure, and it's not going to have the right function. So it's going to be a problem, and you're not going to get the gain that you need for evolution. So again, how do you get this, right? By random mutations over time. How could that generate the complexity and the diversity that we see in the DNA? This is design, this is not disorder. And again, the researchers involved in this said the dual coding functionality represents instead far more sophistication, layers, and dimensions of formal prescription. Tell me how sophistication comes about by random chance, okay? It doesn't. It requires a designer. This is design. This is not disorder. Now, the last hidden code I want to talk about that we sort of know of today, there's probably a lot more we're going to find, and that is something called dual coding genes. Now, this totally goes against everything I learned in college and graduate school, because we always learned that one gene equals one protein. That's the way it goes. As it turns out, that's not really true, because some genes will encode more than one protein. They'll encode two, and there's probably some that encode more, but we haven't found those as of yet. Now, these proteins are very different from one another, structurally unrelated. Okay, so we're not talking about like two versions of the same protein. We're talking about two completely different proteins that do two completely different things. So this is just one example of one called the GNAS1 gene. It has two proteins that are produced from that same gene. And again, one's not just a shorter version of the other. It's actually reading the code differently, which without going into a lot of detail to explain how that's done, but it reads it differently, and so you get a totally different protein as a result of that. And right now they found about 40 candidate dual coding genes, but they use very conservative parameters, so they think there's probably a lot more. This may help explain why we have 20,000 genes, but we have 100 to 300,000 proteins. Because when they initially sequenced the human genome, they really thought they'd find like 100,000 genes, and then they didn't. They only found 20,000. So there has to be sort of some explanation for this, how we get this. And we do see dual coding in viruses. They do this a lot, but because they're like really, really tiny, and so they have to make the most efficient use, so to speak, of their DNA, their genetic material there, and so it makes sense for them, but we've got all kinds of space, right? We have like 3 billion bases, so why do we need to do this? It doesn't make any sense, really, from an evolutionary, at least, perspective. And the researchers involved in this said, historically, dual coding regions were largely overlooked as they violated the accepted views of the eukaryotic gene organization. So eukaryotes are everything but bacteria, okay? Bacteria are prokaryotes. So They actually had known about dual coding regions for a long time, but they thought, oh, well, it can't be coding for two different proteins that actually have function, because you shouldn't see that in eukaryotes. You shouldn't see that in humans. Yeah, well, you do, okay? And they are producing functional proteins that are doing something. Now, dual coding genes, right? Dual purposes, right? Dual things, that's going to be a big problem for evolution. And the authors realized this, of this paper on this. They said dual coding is a costly arrangement because it limits the flexibility of amino acid composition. A silent change in one frame, in other words, one coding sequence for one protein, is almost always guaranteed to be amino acid changing in the other. So even if you get a mutation that might be beneficial to one of the proteins that the gene codes for, chances are you've messed up the other one, okay? So again, because it's coding for two things. Although counterintuitive, they say, okay? This goes against intuition, right? Again, this goes against what we would expect. And the only reason it's counterintuitive because it's inconsistent with evolutionary assumptions that would predict relative simplicity, but consistent with what we would predict starting with God, again, as the designer and creator of our DNA. And the last part of this paper was really interesting because they even had a section saying dual coding is virtually impossible by chance. To reliably find new dual coding genes, we must determine how likely they are to occur by chance. Simulations designed to answer this question show that dual coding is statistically unlikely. Okay? In other words, you've got major problems with this, okay? What are the odds? I mean, so now you don't have to code just for one protein. You got to have the same sequence that's going to code for two proteins in that. Whoa, okay, that's going to be a problem. That doesn't count the duons and the translational pausing codons you got to have too, right? Because you've got all these layers of information there. It's a problem. So yes, it is impossible by chance, and it is statistically unlikely. It's actually statistically probably impossible because it's so low that we can see this has been clearly designed. This is not the result of disorder. Now, the next type of sequence design that I want to address is something called junk DNA, which I talked about at the very beginning. And like I said, approximately 98% of human DNA does not code for protein. And there's different categories of junk DNA. There's what we call non-coding RNAs, repetitive sequences, and pseudogenes. And just to give you, again, a kind of a picture of this, this is a pie graph kind of showing all the different aspects or parts of the human genome. Only that little tiny green wedge there is the protein coding genes. Okay, that's 2%. The other 98% is junk. And as we saw in an earlier video, they think it's leftovers, right, from our evolutionary past, or a genetic fossil, right? Much like you find fossils in the ground, these are fossils in our DNA. And Susumu Ono, who was a geneticist in the 1970s, was the first one to really coin this term. He said, the earth is strewn with fossil remains of extinct species. Is it a wonder, too, that our genome is filled with the remains of extinct genes? So the thinking was that as we evolved up the evolutionary tree, genetic information that was no longer necessary or useful, like information for tail or fins from when we were a fish, began to degrade, okay, and accumulate mutations. But it's still part of our DNA, we just don't use it anymore. Now, from an evolutionary standpoint, this is kind of odd because you would think it would have just gotten rid of that DNA, right? Because if we're not using it, how is it being selected for? Why do we keep it? Because that's how evolution is supposed to work. It's only going to keep it if it's useful. So they would just say, well, we just need more time. That's the key to everything. We just need to have more time, and pretty soon it will go away. But since the release of the human genome sequence in 2000 and 2003, and discovering we only had about 20,000 genes, there's been a lot of interest in studying the junk DNA. And a lot of that research has been done by the group ENCODE. And ENCODE stands for Encyclopedia of DNA Elements. And it involves many scientists and research facilities around the world, so it's a collaborative project. And they have now published dozens of papers in scientific journals. Now, phase one was completed, and it's ongoing, so they'll probably have a new release soon, I'm hoping. But phase one was completed in 2007, and they studied just 1% of the human genome, and they're studying the junk, okay, not the gene. And they found, to their surprise, that nearly all of the junk DNA is transcribed into RNA. And they're like scratching their heads on this, right? Because why would you transcribe it into RNA if it's not going to become a protein, right? Because RNA is that intermediate, so to speak. They didn't expect this. Well, guess what? As it turns out, proteins aren't the only things that have function. RNA has a function, right? It has a function. It's doing something as RNA. Some of the people involved in this research said, we are now seeing the majority of the rest of the genome is active to some extent. It's doing something. It's not just dead, right? It's active. And they may not know yet what it's doing, but they're working on that. Francis Collins, who's now the head of the National Institutes of Health, at that time said there's a lot more going on than we thought. And Eric Green, the scientific director of the Genome Research Institute, said the take-home message is, oh my gosh, this is really complicated. And I always laugh at that because I'm like, yeah, it's really complicated. Okay, a lot more complicated than you ever imagined it could be. I think one of the researchers I had seen at that time compared it to like this giant ball of string that you're trying to unravel and figure out what all the parts mean, because that's how complicated this is. Now, phase 2 was completed in 2012, and they decided to study the remaining 99% of the human genome because the first pilot phase was so successful. And at that point, they termed that 80% of the human genome is functional because it has some specific biochemical activity. In other words, something binds to it, it's being transcribed in RNA. There's activity there. And the third phase of this is to figure out exactly what all that is doing, which is going to take a lot of time and a lot of people to do that. One of the ENCODE researchers said almost every nucleotide, that's a base in DNA, is associated with a function of some sort or another, and we now know where they are, what binds to them, what their associations are, and more. And you got to, I mean, it's maybe kind of hard to understand how big this is. We're talking nearly 3 billion bases, right? And what they do. That's huge to unravel that and figure that out. But they know it's doing something. There's activity associated with all of them. The lead analysis coordinator for ENCODE said it's likely that 80% will go to 100%. Okay, 100% of our DNA is doing something. It's not junk. We've got to come up with a new term for this. I am so tired of calling it junk DNA and putting it in quotes because it's not truly junk. This is designed, right? This is not the result of disorder. Now, in 2012, they did believe, and they still believe this, that most of the functions are regulatory. It's not, some of it might be coding for proteins, which we're still trying to figure that out. There does seem to be indications of that, but a lot of it is actually involved in the regulation, which, like I said, you've got to have a lot of regulation when you have all this DNA in every cell, and you have to regulate it all the time during all phases of life. There's got to be a lot of regulation. And so what's been really neat, though, about this research is that it's really opened the door to looking at variations in junk DNA and how they're related to disease. Because, see, for years, nobody looked in the junk DNA related to disease because they thought it was an evolutionary leftover, right? It's not important. It's not doing anything. And I kind of find it ironic because sometimes people will say, people like myself, being a creation scientist, well, you just believe God did it, so there's nothing to study. I mean, what do you do? And I'm like, God did it, which is why I want to study it, right? It's the result of a logical, intelligent, orderly God. So I have a reason to want to study it and understand it. I believe you can understand it. Whereas if it's just the result of random chance, why bother? But I think, for how many years did these evolutionary assumptions inhibit science, right? Inhibit research because people weren't even looking there. And how many years behind are we on some of this stuff because of those evolutionary ideas? So this is just some of the headlines on this, how junk DNA has been linked to things like cancer, type 2 diabetes, schizophrenia, celiac disease, Crohn's disease, stroke damage. Over and over again, we're seeing that variations there. It's affecting the regulation most likely, and that's what's causing the disease. So it's really exciting to see this and be able to predict and hopefully help deal with these types of things in the future. Now, I want to look a little bit more at the junk DNA so you get kind of a better idea of it and what it's doing. And like I said, there's several types. There's the non-coding RNAs that do something as RNA. They're not becoming a protein, but they're doing some sort of probably regulatory function. Repetitive sequences, you realize that over 50% of your DNA just says the same thing over and over again. And people thought, well, that's obviously not important. It is, okay? And we're going to talk more about that when we talk about the structure of DNA. And then pseudogenes. These are supposedly non-functional copies of functional genes. They look a lot like genes we have, but they don't produce proteins. They don't do anything important. As it turns out, they probably do. And so some of the functions of junk DNA that we found so far, One of the important things is something we call gene silencing. It's turning genes off, okay? Because you got to be able to turn them on, but you have to be able to turn them off. And so it's involved in that, especially the non-coding RNA. Fine-tuning transcription translation, okay? Getting this just so you get the right amount at the right time. Regulating genes during embryo formation. Mouse studies have shown that knocking out, what they call knocking out or deleting certain parts of junk DNA are lethal. The embryo does not even make it, okay? And if they do make it, a lot of times they have severe growth and developmental problems. So junk DNA is important, right? Because if you can knock it out and there's a problem, then we know it has a function. They're involved in DNA formatting, and we'll talk more about that in structural design with the actual structure of the DNA, and in generating genetic diversity, especially in our immune system. So you're exposed to lots of different foreign invaders on any given day, and so your body has to be able to generate antibodies against those, but you don't have all that encoded, so to speak, automatically. You kind of cut and paste, so to speak, gene to be able to do that and form the proper antibodies. And pseudogenes are probably involved in some of the body's ability to do that. So junk DNA is not junk, obviously. It has a function, it's important, and it seriously constrains evolution, right? Because you can't just go making mutations wherever you want and expect everything to be okay. It's not. And just like the hidden codes constrain it. So this is design, this is not disorder. So then, have evolutionists given up the idea that junk DNA is junk? Have they given up on that? And I think some have. I don't think, especially the people involved in ENCODE, I'm not sure how you can keep saying it's junk when you clearly are finding out, observing that it has function. But some of the vocal ones, at least evolutionarily speaking, are still not willing to concede that junk is not junk, okay? They're not willing to believe that. And I want to share with you a couple of quotes from one of them just to illustrate why that's the case. And this comes from John Timmer, and he's a molecular biologist at Cornell and Stony Brook University in New York. And he wrote the following article right after the first release of ENCODE in 2007. And he said, personally, I fall into the it's all junk into the spectrum. If almost all of these sequences are not conserved by evolution, and we haven't found a function for any of them yet, it's hard to see how the none of it's junk view can be maintained. Okay, so here's the problem, though. To say we haven't found a function for any of them yet, yeah, we have. Even by 2007, there had been papers reporting the function of junk DNA. So either he not aware of those, which I find hard to believe, or he's just overstating, okay, this, because we have found a function for quite a few of them, even by that point. Now, what about after 2012, okay? Because that found even more function, right? More biochemical activity. What did he say? He said, and the issue of what constitutes junk DNA is likely to remain controversial. I expect we'll continue to find more individual pieces of it that perform useful functions. Okay, so he's given a little bit, right? Some of it does have function, but he says, the majority will remain evolutionary baggage. Now, or evolutionary leftovers, right? Genetic fossils. Why does he still argue this? I mean, this is stuff we can see right now. We can observe it. It's functional. It's doing something. So why does he still claim that it's not important and it's not doing something? And it has to do with a finding from the 2007 original study, which he talked about in his previous quote. And I want to highlight this part of it now. He says, if almost all of these sequences are not conserved by evolution. Okay, so basically the reason he thinks that junk DNA is still junk, it has to do with an evolutionary assumption, right? Now, what is he talking about? Junk DNA is not conserved. That's what the first ENCODE study found. They looked at mammals. They looked at the junk DNA of mammals because mammals, in their mind, are closely related evolutionarily speaking. They expected the junk DNA to be very similar. Guess what? It wasn't. Okay? About 50% of it was not the same. It was not conserved among mammals. Now let me show you what I mean by that. What is a conserved sequence? So here are sequences, DNA sequences, just a small portion of 8 different organisms. And some of those sequences, as you can see in the box, are identical. Doesn't matter which organism that you look at, the DNA is the same. And so that results in what we call a conserved sequence. That's what we call a conserved sequence. It's the same in every organism, or very similar in every organism that we look at. And evolutionists believe that as organisms evolved, these certain DNA sequences were kept or conserved because they were important, they were functional, okay? So you should see the same. So especially among mammals, you would expect to have a lot of these conserved sequences because they're so closely evolutionarily related. But they didn't find that. And so the people that were doing the ENCODE research said it was the most surprising finding of the study. They said in their paper, however, we have also encountered a remarkable excess of experimentally identified functional elements lacking evolutionary constraint. Okay, blah, blah, blah. What does that mean, right? Basically, we found a lot of the junk DNA is not conserved, okay? It's not the same among mammals. And they said these cannot be dismissed for technical reasons. This is perhaps the biggest surprise of the pilot phase of the ENCODE project. So their biggest surprise wasn't that junk DNA wasn't junk, but that it wasn't junk and it wasn't the same, okay? Because if it's important and it's functional in doing something, all mammals should have, relatively speaking, the same junk DNA, but they don't. So that's what's bugging them about this. It makes no sense from an evolutionary perspective because if it's active, it's functional, it's important, it's doing something, it should be conserved. Because that's how evolution works. It saves and reuses. I mean, when your mechanism is random chance mutation, you don't want to start from scratch every time, so you use what's already there. So all mammals are more closely related to each other than other types of organisms like birds or reptiles in the tree, the evolutionary tree. So the junk DNA should be pretty similar among mammals. Certainly more than 50%, if they know that that junk DNA is important and it's doing something. So the reason they're saying that junk DNA is still junk, right? The whole reason is because it does not fit their evolutionary worldview. It does not fit because it's not conserved among the mammals. So they say, it's still junk. It's just evolutionary baggage, although some of it might have function. The vast majority does not and cannot, because that is not consistent with what we would expect from evolution. Whoa. Okay, now, I'm going to be honest with you. To me, this is probably one of the biggest instances, I think, with evolutionary scientists so clearly denying what we are actually observing in the present because it does not fit with their evolutionary ideas. I mean, this is not a question. It's functional. It's doing something. We're seeing nearly all of it is, and yet they continue to deny it, to deny what is right in front of them because it doesn't fit with their evolutionary ideas. It makes no sense from that perspective. This isn't about the evidence. Like I said, the evidence is clear, but they continue to deny it because of a worldview, because ultimately of a heart issue. And to a biblical creationist, really the ENCODE finding of the fact that junk DNA is not conserved among mammals really isn't all that surprising. And because, again, we don't believe in an evolutionary tree of continuity, so to speak, where we would see a lot of conservation. We believe in the orchard of discontinuity, where you have lots of different kinds, okay, of organisms, but they all have their own DNA, right? They all have their own junk DNA. And while there are some similarities, because, of course, among mammals, our bodies have to do similar things, there's also a lot of differences, and probably a lot of that difference resides in the junk DNA. So what the ENCODE study really confirms is that each animal was created according to its kind with the junk, and it's not really junk, but with the junk DNA that it needed to properly regulate its genes for its kind. And so despite, though, what some scientists say about junk DNA, or some evolutionary scientists, I think there are some, and I think it's increasing because you can't deny the obvious, you can only do that so long, that this is important, it is functional, it is doing something. And I appreciated what these two scientists, who are evolutionists, said regarding junk DNA, which they have studied. They said, our expectation is that one day we will think of what used to be called junk DNA as a critical component of truly expert cellular control regimes. Yes, expert, because an expert designed it, right? God, as a designer and creator of our DNA. This is designed. This is not disordered. Now, we want to talk about structural design. And structural design deals with the actual physical three-dimensional structure of DNA, which is determined by the sequence of the DNA. So sequence relates to structure, as we'll see, and also to chemical modifications that affect how tightly the DNA is wound up in the nucleus of the cell. there is 6 feet of DNA in every nucleus of every cell cell of your body, at least, that has a nucleus. You can fit 10,000 nuclei on the head of a pin. Okay, so we're talking major packaging, right? I mean, major packaging to get that to work. And so it is very highly regulated, because when the DNA is like this, and so it looks like a chromosome, that's usually when the cell is getting ready to divide, the DNA has got to be really wound up, okay, really bound up, so to speak. The blue here is the DNA, and the purple are proteins called histones. They're like little balls the DNA winds around. And so the packaging is really tight here. So when it's like this, you're not going to transcribe or translate anything, okay? It's too tight. It's not available. So to do that, you have to unwind the DNA, which is what you see over here. And when it's unwound like this, then you can transcribe it and you can translate it. So there's a lot of regulation going on to decide how tight or untight, so to speak, that the DNA is. And so there's another layer of information there, right, of regulation and design that's really outside the actual sequence of the bases, but it has to do with the three-dimensional physical structure of the DNA, because this regulates the expression of the genes. So what regulates that? Okay, what controls this packaging? And some of it is the DNA sequence itself. Remember how I talked about repetitive DNA being a type of junk DNA? And these are sequences of bases that repeat over and over again. And there's several different types, okay, like I've talked about before with junk DNA. But again, it's typically been considered junk because it just repeats, right? It's the same sequences over and over again. But nearly 50% of our human DNA is junk. Now, again, we're made by a creator God, and I'm having a hard time believing that 50% of it just is sitting there doing nothing, okay? That's a problem. And so just to show you this again, so each of these bars represents a chromosome, okay, that we have. Each of the colors represents different types of repetitive DNA. And as you can see, in almost every chromosome, we have at least, or maybe a little bit more, 50% repetitive DNA. But that DNA is important. It's important for packaging and getting the right shape and the right structure to the DNA. So it formats the DNA, essentially, because it does have to be wound up, because it is a three-dimensional structure. Certain times you need to unwind it, certain times you need to wind up. So it's involved in packaging it for cell division. Then you need to wind it up really, really tight before it divides. But later, then you need to unpackage it. You need to unwind it so it can be transcribed and translated. Sometimes it's involved in regulation because for some genes, the regulatory element is 1,000,000 bases away. 1,000,000 bases away. So you have to be able to bring those together, the gene and the regulatory element, in close proximity to one another. And that comes from folding up the DNA and bringing those two close together. So there is a ton of formatting that has to go on here that the repetitive DNA is responsible for. I kind of think of it this way. You know how when you're typing, you put spaces between words, right, to make it understandable? This repetitive DNA is kind of like that. It's the spaces, right, so the DNA makes sense. So the DNA either is wound up properly or unwound properly, okay, so that you can get that. The actual sequence may not be so important as it is the number of bases and the location, the actual physical space that it occupied, so you can get the right folding and the right packaging of the DNA. And a good example of this and the importance of the repetitive DNA to packaging and how it can affect gene expression is found in a form of thalassemia, which is a type of anemia, and that's found in the beta globin gene. So beta globin is a gene, produces a protein that's a part of our hemoglobin, which is the protein found in our red blood cells. And the beta globin gene is regulated by a region called the LCR, which is the locus control region. And that region controls the packaging of the DNA and the formatting of the DNA. So it will unwind that part and make it accessible to transcription and therefore translation, so you can get the protein. So here's a picture of the beta globin gene. There's the LCR, that's the regulatory region. There's the gene itself, and there's about 80,000 bases between them, okay? So it's a distance away from it, but it's controlling the packaging of the DNA in that area. And what they have found is that in some forms of what are called beta thalassemia, that LCR is mutated, okay? It's got mutations in it such that it can't package the DNA correctly. It can't unwind it. And so even though the beta globin gene is there and it's intact and there's nothing wrong with it, there's no access to it. It basically winds it up, so it can't be transcribed and it can't be translated, and so you don't get the gene. And as a result, if you don't have beta globin or not much beta globin, you have really small red blood cells and they can't carry enough oxygen, and so you get a type of anemia as a result of that. So the formatting of the DNA is really important to the organization, the physical organization of the genome, because it's important for the proper regulation of the genes. So they're related. So again, how does this pose a problem for evolution? Because how do you explain the origin of these repetitive sequences in the appropriate chance for the proper gene expression by random chance mutations, right? I mean, so not only do you have to have the gene, but you have to have the proper formatting and packaging of that gene in the DNA such that it will be unwound at the appropriate time, transcribed, and translated. So again, there's this whole other layer of information that's there. And again, so it's hard to explain the origin of it, but then you also have to explain how that's going to be maintained, because for evolution to continue, right, you have to generate new genes. And so you also have to get proper formatting of those genes, because to go from one organism to another, you can't just tweak the DNA that's there, right? You got to add stuff, okay? If you're going to go from a dinosaur to a bird, which is what evolutionists believe, you got to add a lot of genes, okay, to get that. And so you not only have to get new genes, but they have to be in a certain location in the genome to allow proper packaging, proper formatting of that DNA. Again, evolution must have got extremely lucky. And so that's a problem, okay, for evolution to continue, because I think a lot of times when evolutionists explain evolution, they make it very simplistic, right? And they just say, oh, you just have to get this gene. Okay, as if that's not hard enough. You've got to do all these other things. You have to regulate it properly. You have to format it properly. And oh my goodness, there's so many other things that you have to be able to do. So there's no way that random mutations over time could have generated this complexity and diversity. This is design. This is not disordered. Now, the next area of structural design that we want to talk about is something called epigenetics and how it has to do with the 3D shape and the packaging of the DNA. Epi means over or outside of, around. So it's not the actual sequence of the DNA, but rather chemical markers and modifications that affect the expression of the DNA. So this is not the sequence. It's something very different. So they basically change the availability and the accessibility of the DNA. They're winding it up or they're unwinding it. But it's not the DNA sequence. So here's the DNA, and it's in the purple or the blue here that you see. These are the histones, okay, that it's wound around. And there are chemical modifications, which are these little tiny colored balls here, that will bind to the histone and affect basically whether the DNA is unwound or wound up. Okay, but they're not a part of the sequence. They're on top of it. They're epi. There's about 12 different chemical modifications that we found so far in human beings. And so it will affect whether a gene is available to be transcribed and translated or not. Now, chemical markers, as it turns out, are heritable. So you not only inherit DNA, but you inherit the chemical markers on the DNA. No one ever thought that. Okay, we used to think that when egg and sperm combined, the DNA combined, all those chemical markers fell off. They don't. Some of them stay on. And it is environmentally controlled, those chemical markers. So it can be controlled by things like diet or stress or exposure to certain chemicals can affect whether those chemical markers are off or on the DNA. And so you may have heard that you are what you eat, but you are also what your mother and grandmother ate. Okay, let me explain that, because every time I say that, I know it just doesn't sound right. What I mean is, okay, they didn't physically eat you, but when your mother was pregnant with you, what she ate affects the expression of your genes, okay? The chemical markers on your gene. And guess what? That child then, when that child grows up, when your child grows up and has children, those chemical markers can still be on there from grandma, okay? So it's passed down, is what I'm trying to say. You're not just inheriting DNA, you're inheriting the chemical markers that affect the expression of the DNA. It can be short-lived, maybe only passed down to one generation, or longer, passed down to multiple generations. So I want to use an analogy to help you understand this a little bit better. I'm going to show you two sentences. They have the exact same words, okay? but totally different meanings because they have totally different punctuation. So, a woman without her man is nothing. A woman without her man is nothing. Now, you can choose whichever sentence you like, but here's the thing, right? The words are the same, but the meaning is completely different because the punctuation is completely different. And it's the same way for epigenetics. You have the same sequence, but it's expressed differently because there's different chemical markers on the DNA. Now, I'm going to show you a short video that explains how this happens. Epigenetics affects the expression of DNA in mice.
The sweet smell of fruit doesn't normally send rats running, but when researchers paired the orange, cherry, almondy scent of the chemical acetophenone with a painful electric shock, lab rats quickly learned to fear it. Along the way, extra neurons sprouted in their noses and in the smell processing center of their brains, making them super sensitive to the scent. This result isn't shocking. What is surprising is that the rats' pups and their pups' pups were also startled by the smell of acetophenone and had the same extra neurons as their fathers, despite never having been introduced to either their dads or the fruity scent before. But how could the pups have inherited something that their fathers learned? Basic genetics tells us that only DNA gets passed along to offspring. Characteristics like memories, scars, or giant muscles can't get passed on since acquiring them doesn't alter the genetic code. But it turns out that instilling fear in the rats did trigger genetic changes, not in the DNA sequence itself, but instead in how that code was read and used in the rats' bodies. In every cell, biological machinery constantly translates DNA into the proteins needed to carry out vital processes. Chemical switches attached to the DNA turn genes on or off or up and down, telling the machinery which proteins to produce and in what quantities. These switches, called epigenetic tags, are why a kidney cell looks and acts differently than a skin or nerve cell, even though all three cells have identical DNA. But the switches in any one cell aren't set in stone. Teaching those rats to fear the fruity smell switched one of their smell-sensing genes into overdrive. Researchers don't know all the places in the rats' bodies where this switch got flipped, but they know it happened in one key set of cells, the rats' sperm cells, which would one day pass along the tweaked genetic material, making the next generation of rats super sensitive to acetophenone. Rodents aren't the only creatures demonstrating this weird type of inheritance. In Överkalix, Sweden, boys who suffered through tough winter famines went on to have super healthy sons, with extremely low rates of heart disease and diabetes. And their sons had the same excellent health, living an unbelievable 32 years longer on average than the grandsons of boys who hadn't gone hungry. To be clear, this does not mean that we should start starving our kids for the benefit of future generations. Scientists don't even know yet exactly which switches the Swedish famines flipped. While we have been able to connect specific epigenetic changes to health effects in mice, we're a long way off from being able to make those connections in humans. That may sound like a bummer, but it's mostly because we humans don't live in the well-controlled environment of a laboratory. And for that, we should be grateful.
Okay. So what we learned from these mouse studies is that the environment can affect the expression of our genes without changing the genes themselves, and it can be passed on to future generations. In humans, there's been a lot of studies trying to understand this better. There's a type of synthetic estrogen that was given to women to prevent miscarriage, and what they found was that in the daughters and granddaughters of those women that took the estrogen, they tend to have a higher incidence of reproductive cancers. Okay, so why is that? Well, it's probably, they're thinking it's linked to epigenetics, okay, something that was passed down. They've also found in twin studies, why is it identical twins, right? They have the same sequence, same exact sequence, but one gets a disease and the other one doesn't. It probably has something to do with epigenetics. They know the gene expression in twins gets more difference over time. There's more differences because they're exposed to different things. They might live in different areas, have different diets. And so we inherit not just a genome, but we inherit an epigenome. Not just the sequence of our DNA, but the physical structure of the DNA as it's altered and modified by these chemical markers. So how does evolution explain epigenetics, or rather, what problems does this create for evolution? Because organisms would have to evolve complex mechanisms to make epigenetic changes to adapt to future environmental challenges, right? Because this is something that's dependent on the environment. So where does that forward-thinking design come from? And according to a recent study on epigenetics and evolution, the people involved in this said this: after the emergence of single-celled organisms some billions of years ago, nature started experimenting with how to diversify gene function without changing the sequence of the DNA. As multicellular organisms evolved, this process of maintenance and function were provided by mechanisms which are called epigenetics. Okay, first thing to note, these are just broad statements with no real explanation as to how this actually occurred, right? But more importantly to note is that nature cannot experiment because experimenting requires a person with a mind, okay? I don't see random, chance experiments happening in the lab, okay? You have to have a mind, an experimenter, a scientist to come in and do those things. Evolution is mindless, and epigenetics is a preparing for the future design. It cannot see the future, therefore it has no way to evolve mechanisms to prepare for that future, right? Forward-thinking design is totally inconsistent with what evolutionary assumptions would predict because evolution works in the here and now. It doesn't have a mind, it cannot see the future, therefore there's no way it can prepare for or predict the future. But it is absolutely consistent with creation because God is omniscient. He designed organisms with the ability to adapt in a fallen world. And he knew Adam and Eve would sin. He knew they would need to be able to do this. So epigenetic mechanisms allow organisms to change easily and quickly in relation to their environment. So if they need to change again in the future, they can do that. Those changes are valuable. They provide immediate benefits for the offspring. They are heritable, but they don't ultimately change the sequence of the DNA. So it's kind of a cool way, right? To change the gene, to change the expression of the genes without making major lifelong, so to speak, changes. So this is totally consistent, forward-thinking design, right? And preparation with what you would predict starting God's word, and God is the designer and creator of our DNA. This is design. This is not disorder. So what I hope you've seen in just these few examples is that the complexity and organization observed in DNA severely constrains evolution from making changes that over time would result in the evolution of one kind of organism into a completely different kind of organism. Nearly all of the DNA is functional and important. It's purposely created and designed. So there's no question that we can know God through what he has created. Romans 1:20 makes that clear. So if design is so clear, then why do many scientists continue to not believe that God is a designer and creator of our DNA? Because they suppress the truth in unrighteousness, right? It's not a matter of knowing because they know in their hearts, but they have suppressed that truth to continue in their rebellion against God. They continue to deny the obvious. So that's why it's so important that in our apologetic, when we defend the faith, that we never forget that this is not about the evidence, because the evidence is clear, but rather it's our starting point, our presupposition, our foundation when we look at that evidence. Do we start with man, who wasn't there during the supposed millions and billions of years of evolution, of Earth's history, or do we start with God, who is an eyewitness to creation and inspired man to write that down? Because God can be known through what he has made, but may we also remember that faith comes by hearing, and hearing by the word of God. You've been a great audience, and I hope you enjoy the rest of your day here. Thank you.